We talked about how planets like Earth form. They start as clusters of stuff. Enough has to hit them to transfer enough heat to them to melt them. They stratify into layers. Takes them a long time to cool down, enough so that there's even liquid water. But as soon as there's water, we have the potential for life. Miller and Urey's experiments showed that the early Earth's conditions were right for producing the stuff cells are made of. And then a sort of a redo of those experiments showed even more things that could come from it. But we don't really have an answer for how we got from those chemicals to the first cells. We have some ideas, is all. We have the idea that microspheres, proteinaceous microspheres, might have been the first baby steps towards cells, even though they're not cells, they can't reproduce, they don't have genetic material. And if genetic material arose spontaneously, RNA probably would have been a better bet for the first genetic material than DNA. It's a multi-purpose molecule. Any questions about any of that?
Going back to cell theory, is this basically saying that all these kind of like the microspheres and like amino acids formed separately, or did they all branch from one singular cell and then that cell divided?
Well, the amino acids and the different chemicals that made up cells certainly would have happened separately. Whether the origin of cells was one protocell or a bunch of little microspheres, we have no idea. Again, it's not something that has been reproduced in a lab situation, so we just don't know. Good, but let's say now we have life. We got to life. We do know that the next step, the next sort of dramatic thing that evolves is photosynthesis. Still in the Archean period. And photosynthesis, of course, leads to something that wasn't there before, and that is free oxygen. Free oxygen, O2, turns out to be a really reactive gas. And it turns out to be a gas that would have absolutely been toxic to most of the cells that lived at the time, as it is toxic to the cells that live at the bottom of hot sulfur vents today. And so free oxygen starting to actually saturate the oceans and then the atmosphere in that order would have caused quite a few changes. Number one, even though most of the iron wound up in the Earth's core, there still would have been a fair amount of it dissolved in the oceans. And what happens when free oxygen, O2, meets free dissolved iron? What do you get? Rust. You get rust, iron oxide, otherwise known as rust. And rust is not soluble in water like pure iron is. And so basically, as the oxygen levels in the oceans increased, all of that iron would have precipitated out. And in fact, we know it did, because you can still find those layers of iron in the right place in the sediment from that long ago. And that would have been when the oceans turned from kind of a rusty reddish shade to their current bluish green. Similarly, oxygen going from the ocean, where the organisms all are, and eventually bleeding into the atmosphere leads to the ozone layer and the current shade of blue that our sky is sometimes when it's not snowing, raining. But for the living things at the time, at least most of them, this was a total disaster. The non-photosynthetic cells that lived at the time, this would have been absolute carnage. This would have been most of them dying. In fact, this is considered the first mass extinction in the Earth's history. Small pockets of those bacteria oxygen was lethal to, found places to hide, right? Wound up in places that were protected and stayed protected. And you can still find them in those places today. But then a very lucky few, we don't know how many, rather than being the ones producing the oxygen, the photosynthesizers, and rather than being killed by the oxygen, there were a few that had the right cellular mutations to use the oxygen, to use the properties of oxygen that make it so reactive. It's electronegativity. Thing loves to grab electrons from stuff, which totally destroys some things. What did certain cells evolve to actually use oxygen? Oh, it's way back. Desperate. What do you mean?
Taking oxygen.
Not turning it into energy exactly. What does the oxygen do when you use it in cell respiration?
Transport chain, electron transport chain.
What does it do?
It takes the electrons and carries it through the end of it.
Yeah, so yeah, to again, not to bring up painful memories, but like you remember this thing and the electron coming in from NADH and boinking along the thing, and we know this one's cytochrome C. We looked at that. But over here, right, something had to pull the exhausted hyperactive kids off the end of the chain. You remember that? And that something was oxygen, and it wound up incorporating electrons and protons and becoming water. This is it. This is what those early cells, it's what allowed them to survive out in the open, is they evolved to use the oxygen. Instead of being destroyed by it, they evolved to use it to pull off their electrons from the electron transport chain and keep it going. So good stuff. It's a good mutation, right? We benefited from that. So I guess what would go in that blank would be final electron acceptor in the electron transport chain. Might be good to write down. I do love a good callback. That was final electron acceptor in the electron transport chain. Almost there. We got photosynthesis. We got cell respiration as we know it. We need internal membranes now. We need about 2 billion years ago. We see evidence of the first prokaryotes that have some infoldings, some internal membranes. Then we get to endosymbiosis. This is about when that would have happened. Remember that endosymbiosis is that process. You might want to look back at this from chapter 7, but the idea that mitochondria and chloroplasts were independent little prokaryotic cells that got gulped into bigger cells by endocytosis. But the little cells were really good at either cell respiration or photosynthesis. The big cells were really good at swallowing stuff. And so they both benefited. Big cell benefited because the little cell produced way more energy than it needed, way more ATP than it needed. The little cell benefited because the big cell was really good at taking in new food for it to digest. And recall that there's plenty of evidence for this theory, that the DNA of mitochondria and chloroplasts, they have their own DNA, looks like old bacteria. They have their own ribosomes that look bacterial. They divide like bacteria, and there's that double membrane thing that would be consistent with endocytosis. Questions about that? You remember that fairly well, maybe? It's a while ago. Yeah, so then 2 steps left really before we're off the races, before we can hit the Cambrian explosion. Sex, which, as we've mentioned many times before, is sort of the fast forward button on evolution, right? Because of the diversity it creates, because of what two things? Crossing over an independent assortment, shuffling up the genes, redealing them every time. More stuff to choose from means natural selection speeds up. That speed up means that some of the colonial cells that liked to live together started to change enough that they became interdependent and couldn't live apart anymore. And that is sort of the definition of multicellularity. And right about then is when we draw the line ending Precambrian time. And beginning the Paleozoic era, of which the first period is the Cambrian period. We call this the Cambrian explosion. It's not a big space impact or a bunch of volcanoes going off. It's an explosion of diversity. It's basically spreading out of life into lots of new niches. As promised, I got you from the formation of the Earth all the way to complex living things. The rest will be your job eventually. Where are we at? Raise your hand if you're done with all the Novolab stuff. What else do we have on our workload? Is that it? There's the shark one. What else? Yeah, I'm gonna look at all that at once. I think she's right. There's one. Oh, cladogram worksheets. Good. Good. I'm gonna lay off today. I'm just gonna give you time to work on Novolabs. No new stuff. Tomorrow we've got a podcast that I'm gonna give you to listen to. Let's go ahead and schedule our tests because we really have one more day and a half left. When's the first ELA? So is ELA Tuesday and Wednesday or Tuesday and Thursday? All right, so your chapter 18 and 19 test will be Thursday. We will be doing other things before then, moving on, practicing for our end of course exam a little bit. You're fine. I mean, you're going to do great, but what's left is to kind of practice the type of questions they ask and learn some test taking strategies that'll be good, not just for this, which is much less important than things like the ACT and stuff. Okay, good. Work on your Novolab stuff. That will be checked tomorrow for a fair number of points. So make sure you get both packets and all the work done. I will leave you alone otherwise.